I am R.KRISHNAN, an assistant professor from St.Joseph College of Arts and Science, Autonomous Cuddalore. Today I am going to present a e-content topic on a classical pathway of complement system. Let's start with some basic introduction about complement pathway and its types. The classical complement pathway is one of the three pathways which activate the complement system. It is a part of immune system. The classical complement pathway is initiated by an antigen antibody complexes with a type of an immunoglobulin such as IgG and IgM. Following activation, a series of proteins are recruited to generate a C3 convertase. The C3 convertase it contains a complement components like C4b2A. The C3 convertase which cleaves the C3 protein and hence they are called C3 convertase. After cleavage of C3 protein, the resulting component have C3b and C3a. The C3b component of the cleaved C3 binds to C3 convertase to generate C5 convertase. The C5 convertase containing C4b, 2a and 3b, which is going to cleave C5 protein. The cleaved products attract phagocytes to the site of infection and tags target cell for elimination by phagocytosis. In addition, the C5 convertase initiates the terminal phase of the complement system, leading to the assembly of a membrane attack complex that can be called as MAC complex. The membrane attack complex creates a pore on the target cell membrane, including or inducing cell lysis and death. So next is the complement cascade pathway. The classical pathway is distinct from other complement pathway in its unique activation triggers and cascade sequences. Activation of the complement pathway through the classical lectin or alternative complement pathway is followed by a cascade of reaction eventually leading to the MAC complex. First is the classical pathway initiation process. The classical complement pathway can be initiated by the binding of antigen antibody complexes to the C1q protein. The globular region of C1q recognizes and binds to the Fc region of the antibody isotypes IgG or IgM. This globular region of C1q can also bind to bacterial and viral surface proteins, apoptotic cells and acute phase proteins. In the absence of this activation factor, C1q is a part of the inactive C1 complex, which consists of 6 molecules of C1q, 2 molecules of C1R and 2 molecules of C1S. So next is formation of C4b convertase. The binding of C1q with the pathogen surface or antigen antibody immune complexes leads to for conformational changes and activation of the serine protease C1R. The activated C1R then cleaves and activates the serine protease C1S. Activated C1S cleaves C4 into C4A, a smaller fragment, and C4B, a larger fragment. So next is the formation of C3 convertase. Surface bound C4b act as a receptor for the binding of C2. The binding of a C2 and C4b results in C2 being cleaved by C1s into C2a and C2b. C2b diffuses into the plasma as a protein inflammatory mediator, while C2a remains attached with C4b, forming the C3 convertase. So this complex containing C4b and 2a. The function of the membrane bound C3 convertase is the cleavage of many molecules of C3 into a smaller fragment and larger fragments such as C3A and C3B. C3A is a smaller fragment of C3, is a potent inflammatory mediator. Then C3B function and structure. The C3B can act as an opsonin. C3B is very similar to C4 in both structure and function. Also has a thioester bond that forces it to attach to the surface nucleophile of the activator, namely the pathogen. Phagocytes have receptors for C3B, and as a result of receptor ligand binding are able to mostly or easily recognize and engulf pathogen molecules, while the anaphylatoxin C3A interact with its C3A receptor to recruit leukocytes. C3B contributes to further downstream complement activation. The final step of this classical pathway is the formation of C5 convertase and MAC. C3B binds to the C3 convertase to form C5 convertase. Then C5 convertase then cleaves C5 into C5A and C5B like C3A. C5A is also an anaphylatoxin that interacts with its cognate C5A receptor to attract leukocytes. Subsequent interaction between C5B and other terminal components like C6, C7, C8 and C9 form the membranated complex, or they can be called a C5B9 complex, which forms pores on the target cell to lysine. So this is the complement classical pathway, how the cascade of reaction will be taking place by the binding of C1 to C9 complement component sequentially, and it's going to create a membrane attack complex on the target surface, example bacteria or viruses. As a result, the cell is going to lyse, and finally the complement components will be disabled. Thank you.